and thank you all for tuning in. Very excited to kick off 2025 with our first NETChatter event. These seminars are held monthly, and we feature different speakers from throughout the world who are either patients, physicians, or researchers, anyone involved in neuroendocrine tumor research. And so for this month, we are super excited to have a talk from Dr. Andrea Gillis. She is not going to be able to be present with us here today. She's a surgeon and she is in the OR currently operating on patients, but she has recorded her talk. So I will share my screen and we can all listen in on that. So the title of her talk will be Exploring the Impact of Race and Adversity on Pancreatic Neuroendocrine Tumors. And a little bit of it about Dr. Gillis. She's a board certified general surgeon. She received her MD from Columbia University and completed her surgical residency at Albany Medical Center in New York, USA. She then completed an endocrine surgery fellowship at the University of Alabama at Birmingham. Her clinical interests include thyroid cancer, benign thyroid disease, hyperparathyroidism, adrenal disease, and surgical treatment of inherited endocrine syndromes, and obviously neuroendocrine tumor research. She's a health outcome disparities and translational science researcher working in the field of epigenetics, specifically examining differences in gene expression in neuroendocrine tumors by race. Her goal is to improve the outcomes of all patients, including those from minority and underserved backgrounds. So with that, I am going to share my screen and we're going to listen to her talk. And I hope you all can see this and hear it. Let me know.
No sound, right? It's here.
Uh-oh, no sound?
Yeah.
All right. How do I fix this?
Huh.
So speakers are same as system. All right, let me press this and see if it fixes it. Okay, I'm going to play it. Let me know yes or no.
I think it's
Did you
still
guys
like we
hear
get a
that?
little bit of I'm getting like a little bit of sound. My guess is that most of it's being filtered out through Zoom if it's playing in the room that you're in. That's my intuition.
You can't. How do I fix this? So I have Elkis here with me. Sorry, guys. One moment. Audio settings?
Can you make sure to share audio
How would I share audio?
as part of the screen share?
Here?
You get share sound.
Share sound.
Yeah.
Okay. All right, let's try it now. I clicked something that said share sound. Tell me if you guys hear it.
And assistant professor and a surgeon.
We got it now? Okay. Thank
With
you.
that.
Okay, all right, let me start over and then onward we go. Sorry, guys.
Hello, and thank you so much for allowing me to present. It's really an honor to be asked to share my work with the NET Cancer Foundation. I apologize that I cannot be there today. I'll be operating, but I'll try to log in between cases. So my name's Andrea Gillis. I'm an assistant professor and a surgeon. I'm in the Division of Endocrine Surgery at the University of Alabama at Birmingham. Today I'll be discussing our work on the biologic effect of ancestry and adversity on pancreatic neuroendocrine tumors. These are my funding sources. So we'll talk about a few things today in the next half hour. So the landscape of pancreatic neuroendocrine tumors in 2025, what we know about how ancestry influences disparities in NET, and the interaction of genomics and epigenetics, as well as social determinants of health and in those social epigenetics, and some future directions. So pancreatic neuroendocrine tumors, although overall rare, have been increasing in incidence over the last several decades. This is our understanding of the overall incidence across different tumor sites, with an update that we published in 2024. And so you can see pancreas here is this teal color. Overall rising, there was this sharp decline that we noted around the time of COVID. And so that's the last year that the data was published. So looking forward to updates to see how things have either bounced back or stayed the same after COVID. So pancreatic neuroendocrine tumors are about 12% of the NETs in the United States. Most are non-functional, but about 20% do secrete some type of hormone that activates within the body to create some sort of syndrome or symptoms. But most are asymptomatic, which generates an interesting clinical conundrum with how to find the neuroendocrine tumors and how to find the tumors that are going to be more malignant and metastasize. About 50% of pancreatic neuroendocrine tumors present already having metastasized. And so we know that obviously the ones that have metastasized are symptomatic or functional or large need to be resected, but there is a little bit of clinical equipoise left up to clinical decision making. Those non-functional pancreatic neuroendocrine tumors that are small, do they need to be resected or can they be observed? So overall, the prognosis is good for pancreatic neuroendocrine tumors, but there are certain patients or subgroups of patient populations that don't do as well. And some of those are the things you might think. So higher grade or more aggressive tumors, tumors that have already metastasized or moved outside of the organ of origin. Interestingly, women do better than men, but there's certain social and demographic factors that cause worse outcomes, or linked to worse outcomes. So patients who are uninsured, patients who are single versus married, and patients who identify as non-Hispanic Black versus their white peers, when controlled for all these other variables, still do worse. And so that's what our lab studies. Why are these patients more likely to die from neuroendocrine tumors than their peers? So we know racial disparities in outcomes exist across many cancers, and there's likely a combination of factors that leads to this. So socioeconomic status or issues of access, comorbidities, other biologic factors that place certain patients at higher risk, and then health system and environmental factors that increase minority morbidity. But when we look at neuroendocrine tumors, we don't think that access explains all of the differences that we're seeing. For example, we know that Black patients with neuroendocrine tumors have a lower overall survival, but they may present with tumors that are smaller and even metastasize at smaller sizes. So we saw that in our local UAB data, and we decided to validate this and a larger data. So this is the UAB group's query of a national cancer database, as well as a NET-specific database, the US NETs group. And both of these corroborated our anecdotal data that Black patients are more likely to present with smaller tumors. And when they present with these small tumors, they're more likely to have already metastasized compared to their white peers. And so that makes you think it's not an access issue, because these patients were able to both access a surgeon and have surgical resection at a small size. So the tumor, we think, wasn't delayed in diagnosis, but the biology was already more aggressive. So is it a biologic difference in addition to or instead of an access difference? And so we know that there are lots of other things in consideration. So others have shown that even when Black patients do qualify for surgery, they are less likely to get that surgery. And when they do get surgery, these sort of differences in outcomes do get better. So is it access or biology? And, the lab is interested in trying to answer this question. So another look at pancreatic neuroendocrine tumors, specifically in the SEER data set, looking at overall survival, we tried to tease apart this access versus biology question. So we looked at the last 20 years in pancreatic neuroendocrine tumors, how patients are doing. So we noticed a few things. In the later years, patients are surviving more often. The racial difference, although there, was not statistically significant. And this is when controlling for other demographics. Patients who present with more distant disease, again, more likely to have lower survival. But then these social differences also rose to the top as influencing outcomes. So patients who live in neighborhoods that overall make less money or live in poverty, worse outcome, and patients who identified as single versus married, worse outcome. So these are more social factors that seem to be influencing neuroendocrine outcomes as much, if not more, than the racial or ancestry factors, but it seems to be a multifactorial thing. So one way that our lab is looking to analyze how these different factors influence tumor biology is with the genome. And so genomic changes are the changes in the expression of a gene. So the sequence, the actual C's and G's, don't actually change in the genome, but something happens in the genome, in the tumor, that changes how the genes are expressed or translated or transcribed. And so our lab is trying to connect race and ancestry as well as adversity to these differences in gene expression. And a lot of this has been explored in the neuroendocrine world, many of you may know, and it's becoming more and more common because these sequencing technologies are becoming cheaper. So we know pancreatic neuroendocrine tumors have numerous epigenetic changes. It's mostly loss of function changes or mutations, meaning something that protected the tumor from developing or metastasizing in the genome now goes away. And so several groups have looked at this and even quantified the specific genes that are differentially expressed in neuroendocrine tumors that either predict outcome or development. And so commonly altered signaling pathways include DNA damage repair, chromatin remodeling, telomere maintenance, and mTOR activation. So this has been studied decades ago. This is kind of well established and accepted in the world of neuroendocrine tumors. However, a lot of these studies were done in either race agnostic, meaning they didn't report the race of the patients, or they did not include a variety of races in the experiments, or they just didn't know, because oftentimes in these big data repositories of genomic information, race is not included as a demographic, or at least it wasn't in the past. And so our lab has actually tried to quantify this. So we know the US population is about 60% white, now closer to 55%. This is from 2020. So this huge swath of patients identify as non-white. When we look at cancer genomics as a whole, all cancers, in this 2018 paper, only 9% or so of all patients who had their tumors sequenced and reported and published were non-white. In our lab, Dr. Herring has looked at the number of manuscripts specifically in pancreatic neuroendocrine tumors reporting race that even bring up race, and the number is staggeringly small. And then he looked at specifically the number of studies of PNETs by race, and again, only 1.3% of patients studied with PNETs report race. And, you know, at UAB in Alabama, we have a unique situation where almost 30% of our patients identify as Black, which is overrepresented compared to the national database, the national census. So that makes us question or worry, how representative are these epigenetic findings in relation to our patients that we are seeing every day? And so our lab is trying to rebalance this in order to understand the influence of race and ancestry on tumor biology. So I just want to take a second to discuss definition. So we are trying to understand genetic variation in the context of health disparities, those differences in outcomes that I presented earlier. When we talk about ancestry, that's a biogeographical definition to describe where genes come from. So, you know, are your genes more from your European ancestry or your African ancestry? When we describe race, that's more of a social category. That's what we use to describe health disparities within societies by racial categories, which may not be necessarily aligned with ancestry. And then when we talk about ethnicity, that's more of a cultural description. So traditions, lifestyles, diets, and values that go along with the way people live their life. So at UAB, we are trying to understand the drivers of disparities in pancreatic neuroendocrine tumors, specifically for minority patients. So when we look at all of our patients who have had tumors resected for pancreatic neuroendocrine disease, we've had about, it's now closer to 200 patients over the last 20 years or so have had surgery for pancreatic neuroendocrine tumor. It's important that they have surgery because we need some of their tissue in order to sequence and answer this biology question. So these are the demographics of our patients. So the Black patients are younger, they tend to have tumors that are larger, and again, these social factors, more likely to be single. We do not see, a large difference in the outcomes. A lot of that has to do with because it's only 200 patients, so it's a small data set, and because neuroendocrine patients tend to do so well. But there are some trends that we're starting to see. In order to sequence these patients and understand their epigenetics and their transcriptomics, how the genes are expressed, we have to physically take their tumors and send them off for sequencing. So we do both RNA sequencing and DNA sequencing to get the transcriptome and the methylome reported back. And so here's a picture of a paraffin-embedded slide of a pancreatic neuroendocrine tumor. And so you can see this outlined area that's also expressing synaptophysin here is where the pancreatic neuroendocrine tumor is. So we try to take just that tumor region and sequence it to get the following data. So here is the work of the MD PhD in our lab, Dr. Herring, comparing the gene expression of Black patients compared to their white controls. And so you can see off the back, this is a volcano plot displaying differentially expressed genes. So the genes further to the right are enhanced in expression. The genes further to the left are suppressed in expression. And then any gene above this dotted line was statistically significant. So there are hundreds of genes, or maybe 800 genes, that are differentially expressed. And so that's a lot of data, but it's also novel data that we didn't know, that these genes are differentially regulated or expressed in minority populations. And so what do these genes do? Dr. Herring did a gene sequence expression plot. So this shows what pathways are these genes on the left most likely to reside on. And so the number one activated pathway was in the immune system, so humoral immune response. And so I'll talk about why I think that is in a second. So in order to narrow those sets of genes, we took the genes that were linked to outcomes, so clinically relevant differences. So genes that predicted a composite metric of either advanced disease, meaning you presented with metastasis, or you recurred or progressed after treatment. So we used this composite metric to narrow down the genes to the clinically relevant genes, and we got about 30 genes. And then on top of that, we've been exploring how to map ancestry onto these patients. So a patient may self-report or self-identify as Black or white, but they may have a different percentage of actual African ancestry or European ancestry that may skew gene expression. So we're trying to look at race more as ancestry, which is a continuous variable instead of a yes/no. And so when we did that, about 19 genes came back as both clinically relevant and differentially expressed. So now we're looking into these 19 genes to biologically validate them in tissues and in our tumor specimens. So that's our gene expression analysis based off race. But remember, I told you there are multiple things at play here, not just self-identified race that predicts inferior or worse outcome. So there were also those social factors that I showed you before. And so in order to place this in a broader context of social determinants of health, which are the conditions in which people are born, grow, live, work, and age. I reached out to one of our local experts, Gabriella Oates, who is a sociologist, has a PhD in studying these specific conditions and how they impact disease, cancer, and outcomes. And so when we look at these different variables that can affect outcomes, there are tons of them, and there are lots of different ways to break down these different domains. But overall, you want to look at the wider set of forces in the environment, in the government, that affect how well a patient is going to do that has nothing to do with their disease. So there are these structural determinants, things like socioeconomic position, how much money you make. And then there are the intermediate determinants. Where do you live? How well do you handle stress and disease, the things that you use to recover and recoup? All of those things play a role in health and overall well-being. So when trying to decide how this affects gene expression, we need to find a way to quantify this. So this is a very complex but more accurate system of social determinants of health. This is a little bit more simple. So this is from the CDC, Healthy People 2030, that acknowledges these social determinants of health and has come up with 5 specific domains that can be measured in order to understand a patient's exposure to adversity. And so these 5 domains include education, healthcare access, the neighborhood, social and community context, and economic stability. And so we can try to measure each of these domains by pulling out publicly available data at the US Census level. And so these are the specific metrics that I've used to measure each of the 5 domains. And so we look at how our local patients fit into each of these metrics and compare them by race. And so I've taken the patient's billing addresses, mapped it to their US Census tract, which again is publicly available data, and we can measure the cohort as a whole and their level of adversity. And so you can see at every level, our minority patients experience more adversity. So lower income, lower percent of graduation, increased percent of poverty, more people live in the neighborhood are uninsured, and worse vulnerability. And so now we're trying to map those measures of adversity onto tumor biology. And so we're definitely not the first to do this. This is, again, a growing field called social genomics, where we're trying to measure how patients differentially express their genes based off of their environment. Things like family history, obesity have also been studied. And so the idea is that we can measure things that influence outcome beyond access. So how does actually living in a neighborhood that is impoverished affect your biology, including your tumor biology, in addition to whether or not you have access to a surgeon or a high volume physician? Some other examples of people who've done this really well are mostly in the benign world. So here are a couple of examples. This one is looking at poverty specifically and how the intersection of sex, race, and poverty causes differential expressed genes. This is in blood samples. Here's another example of a measure of economic hardship and how that affects gene expression with an outcome called epigenetic aging. So beyond your chronologic age, there are different epigenetic clocks that can measure how quickly your cells are aging. And so this study showed that in patients who live with more economic hardship, their cells have aged a lot faster. So and this one is looking at measure of subjective discrimination. So patients who have experienced discrimination in their life, measuring how their genes are expressed or differentially methylated. This shows, each chromosome and which genes are more highly methylated based off of exposure to discrimination. So these are, benign diseases looking at how the environment and your social situation affects your disease or your biology. So now we want to take this to the cancer stage. And so a good framework that I use to think about this is those predisposing factors or social determinants or adversity cause something to happen within your body. And usually, or one hypothesis is that it increases stress. And stress is also known as allostatic load. And so your body has to constantly respond to that stress. And that causes different systems, different hormones in your body to rev up in order to respond to stress, almost like, you're running from a lion in the jungle. And so when those different systems are revved up, that has in-organ changes. And so that can cause metabolic changes or increased cachexia. That can cause cardiovascular changes, raising blood pressure, decreasing heart function. And then all of these things, these secondary outcomes, then reduce survival via morbidity and mortality. And so you can think of this as affecting cancer development as well as a secondary outcome. And so the lab is interested in finding out what's at these arrows, what is mediating the effect of these predisposing factors to reduce survival or increase disparities. And so we've taken each of those 5 domains of social determinants of health and linked them to gene expression. So when we've measured those billing addresses and linked them to census tract, we can say this person lives in a neighborhood with low income. And then we look at those patients and see how their genes are differentially expressed compared to those who live in neighborhoods of high income. And so we've done this with each of the 5 domains, but in the interest of time, I'm just going to show you the income domain. And so you can see there are several genes, both enhanced as well as suppressed, differently based off of income. And so some of these genes have really interesting functions. So on the suppressed side, this KIF18A has to do with cell transport. This RAL-GAPA has to do with immunoglobulin genes. So again, related to what we found with our ancestry analysis, may link to the immune function. So something going on with the stress response affecting the immune function. And then on the activated side, this ETNK, the link gene, this top link gene has been linked to prostate cancer and liver cancer. And then this TICAM gene has been linked to differential metabolism and toll-like receptor pathways. So all of these genes are now being explored with the hope of future biologic validation, which is now underway in the lab. When we look at the pathways of those genes, again, the enhanced pathways have to do with metabolism, perhaps that stress response, and then the suppressed genes have to do with the immune function. Perhaps this is an exhaustive response, so the persistent stress response, enhancing the immune pathway is causing a burnout, and that's why we're seeing a suppression in that pathway. Those are some of the hypotheses that we're working out now. We've also looked at the data by methylation. So perhaps differential methylation is driving the different gene expression. So this is a Manhattan plot where we look at every chromosome to see which genes are differentially methylated. And so we have a whole new set of genes that we're exploring to see how that methylation difference affects gene transcription and expression. So part of the biologic validation is then taking the differentially expressed genes that we found on the sequencing data back to the tissues that we have to validate, well, are we actually seeing the protein that's encoded by that gene differentially expressed in the tissues themselves? And so the tech in my lab now, Stuart, has been going through our tissues that we've collected from surgical resection and checking them for immunofluorescence to see, does that gene that we found match up with the protein expression? And so he's been painstakingly going through all of these genes that we've been identifying. So a lot more work to do. This is just the beginning of a really exciting new field of cancer epigenetics and social epigenetics in the context of neuroendocrine tumors. And so I think this has a ton of potential both in examining biomarkers. So if the patient gets a biopsy or even a tumor resection, can we use these differentially expressed genes to then move forward and predict how well patients will do? Can we develop drugs that can target some of these genes? Can we learn more about the tumor heterogeneity and the microenvironment by seeing where these genes and these proteins are enhanced or suppressed within the tumor? And we can also broaden this to other cancer types. I'm particularly interested in pancreatic ductal adenocarcinoma. And I think it's really important to keep patients and their families in mind as well. As we are learning more about epigenetics and the impact of ancestry and adversity on tumor biology, making sure that we are educating patients about their risks and how to interpret their risks based off of these findings. Of course, they need to be validated a lot further, but in the future, this is something that we're going to have to be working with patients on and making sure they understand all of this data that we can generate from sequencing a tumor. So thanks again for your attention. Of course, I want to thank all of the patients that contributed to this work, as well as my clinical partners and research partners. Really, I couldn't do any of this without a lot of help. And so I just want to thank everyone and also thank our funding sources for being able to complete this work as well. Please reach out to me if you have any questions.
All right. That was a phenomenal talk. Oh gosh, it's going again. So actually, we have Dr. Gillis here with us. I think I saw her. Oh, she's in the OR. Oh my gosh.
I'm here.
She's here. Thank you for tuning in. Dr. Gillis, do you have a moment for questions or do you need to get back to what you're doing?
I've got some time.
Okay. Does anyone, I'll open the floor, have any questions for Dr. Gillis with her presentation about social and racial influences on pancreatic neuroendocrine tumors? Definitely an exciting area of research. John, yes.
Hi. It was really interesting to see that. And I really appreciated the way that you laid out the differences. You making it really clear what you're studying at the different levels and how you're layering that on to create a picture, and then possibly even do some implementation to perhaps change outcomes.
Yeah.
So that on that note, because there's lots of different ways that you could try to do that. But what I'm sure you've thought of a million of them. One I thought of that I wanted to ask you about was since you are seeing these differentially expressed genes that might that you have correlated at a macroscopic level to stress. This is okay. I guess it feels super wacky to me, but I'm going to say it anyway. What about in the long run, as you go on to show that these really do seem to be meaningful, maybe that could be used to justify a study that might even try to look at how they would change as you change the environment somehow. Like we can imagine an integrated cancer center, which these already exist, that includes, say, psychiatry and therapy. Because some of the things, even if you remove the current stressors, it'll still leave lingering trauma, if you will. And so having those integrated services, which some centers have, but maybe it's tough to justify, maybe insurance doesn't cover it. And so then you can show that there's an actual difference, then maybe people will pay more attention to that. And it won't just feel good, but you'll say, look, it feels good and it's the right thing to do. And we really have the data to show it.
Yeah,
So,
what a great question. Not wacky at all.
Okay.
Yeah, and I think that kind of is at the crux of doing this work. So, I wanted to move away from just looking at race and ancestry because those are things that are pretty hard to change in terms of reversing them. But, the goal is to try to find targets that we can develop drugs for those immutable things. But in the world of like social determinants of health, adverse environmental exposures, we think a lot about, what are we going to do if we find these things?
Yeah.
So if someone tells you, yes, I can't afford my medication, do we actually have resources that can help them? And then
Right.
you think about the downstream stress of everything in their world and their environment that leads them to not be able to afford medications, for example. So yeah, and I think there is some, not a ton of work being done looking at reversing epigenetic changes. So one example that comes to mind is smoking cessation.
Yeah.
And so there have been studies showing that, when you test sputums, test blood in patients who have stopped smoking, some of those negative changes are reversed. Not all of them, and we don't know how that affects their overall outcome. But I think that's some exciting frontiers that we really should be pushing and thinking about, especially in the social determinants world.
I meant to add this, is that, of course, at a base level, just making sure the patient can make it to the center for their scans to see the doctor. And then again, trying to justify a center, like, look, if you really want better outcomes for your patients, like, you invest here,
Yeah.
and this will make a difference. Instead of investing in other places that they might invest. I mean, who cares if you have the latest, I mean, you probably care, right? But who cares if you have the latest and greatest surgical equipment if the patient can't make it and they're going to have a poorer outcome because of that. And I bet a service to get the patient costs a lot less than some equipment. I know Linacs, they're very expensive. MRI, very expensive.
Yeah. Very good point. Now you're talking like systems changes, like even beyond the hospital level, the state level, the country level. Like, how can we really bring up our most vulnerable patients? So I like where your head's at. I don't know if that answers the question, but I asked the same question myself.
I know. I think it did. You're thinking it kind of like that. The way that I'm thinking about that isn't super crazy, and you're thinking along. You're thinking, yeah, that is the kind of way that you see this applied. I guess that was really my question. How do you apply this?
Yeah,
that's a great question. Maybe one day, John, we can have a conversation on how we're going to integrate mathematical modeling into this research. Any other questions? Well, we've got Dr. Gillis here I'd say in the flesh, but we're virtual flesh. All right. Well, with that, I will go ahead and say thank you again, Dr. Gillis, for sharing your research with us. We're very fortunate, and that was an excellent presentation discussing a novel and upcoming important field in neuroendocrine tumor research. And a big thank you to everyone who attended this talk. And Selena, do you want to say anything else before we close?
Just that thank you very much, Dr. Gillis. And our next meeting, which I hope you can all come, it will be Thursday, 27th February, same time. And I can't remember who's speaking. Who's speaking, Rachel?
I don't remember either, but we will let you all know who is speaking. It'll be someone great.
It is already arranged, and I can't remember. But so it'll be a surprise. Those of you that are not on my email list, if you want me to email you the links, then please do email us. So it's contact@netcancerfoundation.com. And we can add you to email list so you get the invites and the links. We don't harass you. It is only once a month. You won't get bombarded with emails from us. So it is just the links for the meetings. So thank you very much, everyone, for coming, and hopefully see you all again next month. Thank you, Rachel.
Thank you all. Bye-bye.
Thank you very much.